Welcome to the lecture about DNA replication. But before we start talking about how DNA replicates, we're going to review what DNA is, where it was discovered, and how it is structured. So DNA is also known as deoxyribonucleic acid. That's the fancy word for it, deoxyribonucleic acid. We shorten it to say DNA because it's a very long term, but this term actually tells us a lot about the DNA molecule. We can see that it is a nucleic acid. We can also see that it has a deoxyribose sugar, which will come important later. We know that DNA carries the genetic code, which stores and transmits the genetic information from one generation to the next. So DNA is transmitted from your parents onto you. We know that DNA is made up of nucleic acids. You remember nucleic acids from Unit 1. We're coming back to that. And we know that chromosomes, these guys here, these big X's, are made up of DNA, and they're located only in the nucleus. So we know that DNA is located in the nucleus of eukaryotes. So where is DNA? We know in eukaryotic cell, it's found inside the nucleus, so all the DNA can be found here. For prokaryotic cells, it just floats around in the cytoplasm because they do not have a nucleus for it to be in. So what is the function of DNA? DNA contains the instructions for making proteins. So the DNA here has a code within it that tells our cells to create proteins. And when those proteins are created, they in turn express genes or traits. So you having blue eyes or brown eyes is actually the creation of a protein that allows your eyes to be that color. And that protein is the instructions to make it is found in our DNA. We know that all living things contain DNA, and they also contain the same parts that make up DNA. So if you remember those nucleotides from our first unit, all living things have some sort of DNA made up of nucleotides. Now, the order of those nucleotides are going to be different depending on the living organism, but they all contain those basic parts. Now, who discovered DNA? There's three famous scientists that we like to look at for who discovered DNA. The first two are James Watson and Francis Crick. They are the first people to create a model of DNA. They found that it was a double helix, and they created a physical model, and because of this they received a Nobel Prize in 1962 for their work. But they created that model from Rosalind Franklin's work. She's the first person to take a picture of DNA using X-ray chromatography, and this is her picture here. This is the X-ray chromatography from Rosalind Franklin. This is as if you're looking from the top of the DNA molecule, so this would be the outer blue areas here, while these in here are showing the nucleus or the nitrogenous bases in here. So from this they found that they were ordered in such a way that it was a double helix. And so that's where we got the double helix from. So let's recall the structure of DNA, right? We know that DNA is the polymer, right? The big piece made up of multiple monomers. And it's made up of those individual nucleotides, and those nucleotides are those monomers. We know that a nucleotide is made up of one phosphate, one sugar, and one base. That creates that one nucleotide, just right here. We have that phosphate group, that sugar, and the base. And we know that nucleotides are the building blocks of DNA. Remember earlier when I talked about that deoxyribose? This is specific to DNA. This sugar group is a deoxyribose sugar, which is specific to DNA. It is different in the compound RNA, which we're going to talk about in our next unit, which only has a ribose sugar. And so that deoxyribose tells us that it's this type of sugar connected to a phosphate connected to a nitrogenous base. So, when we're looking at DNA, when we look at the sides of the ladder, or the parts of the ladder you would put your hands on if you were to climb it, they have phosphate groups and sugar. So it alternates phosphate, sugar, all the way down that model, which is this portion of the DNA molecule. So these are just repeating phosphates and sugars. While the steps of the ladders are the nitrogenous bases. And so you can see these nitrogenous bases, the C, the G, the A, the T. These contain nitrogen, which is why we call them nitrogenous bases. And that is where you would physically step if you were to get on this ladder. These nitrogenous bases are what codes for your traits. So the different order of these nitrogenous bases are what's coding or giving the instructions for the traits. Since these sugars and phosphates are just repeating units, they're not the actual code. The code is found in the center here with these nitrogenous bases. The 4 nitrogenous bases are adenine, which we use an A to show that in short form, thymine, T, cytosine, C, and guanine, G. So you'll see these 4 nitrogenous bases alternating within that DNA molecule, and the order of those, once again, are what code for the traits. Now, how do we know how these bases match up with one another? Erwin Chargaff, a scientist, analyzed the relationship between the DNA nucleotides. And in 1947, he reported that the percentage of cytosine in a DNA molecule equaled exactly the same amount of guanine in the DNA molecule. And they also noticed that the amount of thymine in the DNA molecule equaled the amount of adenine in the DNA molecule. So he concluded that adenine must pair with thymine because they have the exact amount, while guanine pairs with cytosine as they have the exact amount. These are the pairing rules. So if I were to give you a segment of DNA, the first half, you should be able to find the second half by remembering their pair. So if I gave you an A, you should know that it pairs with a T. If I give you a C, you should know that it pairs with a G. Okay? One way to remember this is the term apples are tasty and cookies are good, or apples go in trees, so A goes with T, and cars go in the garage, C goes with G. This is just a picture of that. A's go with T's, C's go with G's. So let's practice. If there are 22 thymine bases found in DNA, how many adenines should there be? Well, since thymine pairs with adenine, there should be 22 adenines because they are partners. If there are 45% guanine in a DNA molecule, what other nitrogenous base would be found at 45%? Well, what pairs with guanine? The cytosine. So you would see 45% cytosine there. So now that we know the structure of DNA, what it looks like, how they pair, now we're going to look at how DNA replicates. So, well, why would we need to replicate? When would your body need to make more cells or DNA? DNA needs to be replicated so that there is an exact copy of DNA for each new cell that's created. So when we make new cells, whether it's repairing things in our body or we're growing, we need to have an exact copy within those cells. And so we have to copy that DNA in order to put new DNA in those cells. So in order to make new cells through cell division, DNA in the parent cell must be replicated before the cell division so that the new cell has a copy. So how is it done? Well, the first step, the replication begins when the DNA unwinds and unzips, specifically by this helicase enzyme. There's an enzyme here that goes in and it will unwind the DNA to make it flat instead of in the helix, and then it will unzip this DNA so that you can have the two portions of the DNA apart from one another. And then each of those strands will serve as a template. So you see here, here's the original strand. It originally was in a helix, but then it unwound, so it's flat. Then this helicase enzyme will come through and break it apart so that each of these become a template. This is step 1. So once again, that DNA helicase enzyme will go through and break apart those pairs and unzip between the two bases, and it creates two complementary halves. Then step 2, one nucleotide at a time, DNA polymerase will move along the parent strand, constructing a new complementary strand. So now that these are apart, they're now going to be used as the template to add more nucleotides onto each side. So they know there's an A here, they added a T. There's a C here, they added a G. That DNA polymerase is coming through to add those nucleotides back on. So free-floating nucleotides, which are just free-floating in the nucleus, will match up to the original strands by DNA polymerase. So once again, this enzyme here, DNA polymerase, is coming through and attaching new nucleotides on each side. You will need to know how to add those nucleotides on. So if I asked you what would be the other side, what side would DNA polymerase put on, you should know once again that A pairs with T and C pairs with G. Then for step 3, the nucleotides are connected to form a sugar phosphate backbone. Enzymes will proofread the DNA and repair any errors, because if there are errors, you don't want to put that into a new cell. You need to proofread it first. And each new molecule is now made of a part of the original chain and a new chain of nucleotides. So now, because it's been separated and new nucleotides have been added on, you have 2 sets of DNA here. One side is going to be the old strand, the other side is going to be a new strand for each of them. So each of them is going to be half old, half new. And then once the replication is complete, the cell can begin dividing again. So once again, 2 new strands are created, half original, half new, because they're going to bond together in that step 3. Really quick before we end, we're going to talk about enzymes again. We remember enzymes from our first unit. It's a protein. Those proteins specifically speed up chemical reactions in an organism. And so they're doing some type of job to complete a task in an organism. And there are 2 main ones that we used in the DNA replication. Remember helicase from step 1, unzipping that DNA and pulling it apart. You need to remember that enzyme. And then also DNA polymerase, which is building the new strand. So that's the one that's bringing in the new nucleotides and putting it on the old strand of DNA. Before we go, we're going to add some bases on this, right? So what's going to pair with A? T, right? So the other side here would be T. What pairs with C? G, so you would put a G here. What pairs with G? C. C pairs with G. A pairs with T. A again pairs with T. G with C, and T with A. So if I asked you to be DNA polymerase and add the second half of this DNA molecule, you should, from those base pairing rules, be able to find the other side of the molecule. So that's all for today. You know the structure, you know how it replicates, you know where it comes from, and where it resides.